Good morning. Today I'll be giving a brief overview of the FDA genomic tool ArrayTrack. ArrayTrack has been developed at NCTR as an integrated solution for microarray data management, analysis, and interpretation. Apart from microarray data, ArrayTrack also allows for integration of data from other omics platforms and also incorporates study data from both clinical and non-clinical studies. Now, ArrayTrack is popular among all the FDA centers. In fact, it's the key review tool used for the FDA Voluntary Genomics Data Submission Program. It's also been used in the Microarray Quality Control Program, and over 100 FDA reviewers and scientists have been trained to use this tool. So apart from its popularity among the FDA centers, ArrayTrack is also freely available to the public, and it's become very popular in different bioinformatics courses in universities, for example in Colorado State University, Georgia Tech, University of Arkansas, Little Rock, so forth. We've given training courses all over the world in countries in Asia, Europe, and such. And ArrayTrack has also found its way into the private sector. Recently, pharmaceutical company Eli Lilly, after a rigorous due diligence testing, chose ArrayTrack as their primary microarray data management and analysis tool. I briefly mentioned the Voluntary Genomics Data Submission Program. That's actually a novel data submission mechanism which encourages sponsors to interact with FDA by submitting genomic data on a voluntary basis. Now, this is beneficial to both the FDA and to the pharmaceutical companies because it helps in developing best practices on how to perform pharmacogenomic analyses and also how to regulate pharmacogenomics data submissions. Now, VGDS recently got renamed as VXDS, standing for Voluntary Exploratory Data Submission, because it's been expanded to handle not only genomic submissions but also proteomics, metabolomics, and SNP data submissions. So in order to handle all these types of data, VGDS required a strong bioinformatics infrastructure, and ArrayTrack has successfully filled that role. ArrayTrack has a 7-year history behind it, and during these 7 years it has experienced tremendous growth. When the first version came out in 2001, it was just an in-house data management tool mainly for filter arrays. In 2002 we came out with version 2, which was able to handle customized 2-color arrays as well, and in late 2003 ArrayTrack was made open to the public. In 2004 is when the VGDS program started, and as I mentioned, because of VGDS requirement for a strong bioinformatics tool, ArrayTrack also had to be expanded to meet these requirements. The major change in version 3.1 was that ArrayTrack was expanded to handle Affymetrix gene chips, which means that it was able to directly load cell files, convert cell files to probe set files, and so forth. In 2005, the MAQC program was started. This is a program that was an FDA-led collaborative effort in order to come up with some kind of a consensus solution regarding the quality of microarray experiments. MAQC required a bioinformatics tool that was able to handle 7 different commercial platforms. So ArrayTrack had to expand further. And we came up with a generalized data management scheme to handle most, if not all, commercial array platforms. And between 2006 and the present, we've come up with 2 different versions, 3.3 and 3.4. And this has been majorly due to the expansion of VGDS program to handle proteomics, metabolomics, and study data. In order to handle study data, ArrayTrack has been incorporated with the SimpleTox format, which uses the SEND standard. I'll go into that a little further, but that's the major improvement in 3.3 and 3.4 versions. This picture shows how integrated ArrayTrack is. It is not only capable of handling microarray data, but it's also can be used to do meta-analysis with proteomics and metabolomics data. It can definitely store any type of study information for clinical and non-clinical studies. And we also have public data which has been pulled from public repositories, and it's been stored in ArrayTrack. This is very useful for biological interpretation. This is the architecture of ArrayTrack. It is a client-server system. So on the bottom tier you can see the server, which is predominantly databases to store the microarray, proteomics, metabolomics experiments, to store the study data, and to store data that's been collected from public databases such as pathway information, gene annotation, protein, ortholog information, so forth. And the client tier pulls the data from these various databases, and it uses that to perform analyses and different visualizations, and this is what the user is able to view and access. These two charts just show how ArrayTrack is becoming more and more popular. Both the web access form of ArrayTrack and the local installation, the number of users has just kept on growing over the years. This slide shows a sample dataset which will be used in the next short tutorial of ArrayTrack. This dataset consists of primary rat hepatocytes which have been exposed to cadmium acetate. It's using an AFI chip which has 1030 genes, and what we're trying to do in this experiment is compare 12 different hybridizations, 6 treated versus 6 control. And we're using a standard dose and a 12 hours time point. And what we're trying to find is the genes and the pathways which are differentially expressed or dysregulated between the treated group, the 6 treated samples, and the 6 control samples. I briefly mentioned the SimpleTalk standard, which is a standard used for data import. It allows researchers to not only import their expression data, but along with that import their study data, which means that they can store animal information, toxicological endpoints, clinical endpoints, and so forth in ArrayTrack along with their expression data. And this allows us to capture essential information related to a toxicogenomic study. It allows us to do cross-study analysis, and this is compliant with the MIAM and SEND standards. It's basically an Excel spreadsheet with required and optional fields, and the researcher would have to fill this spreadsheet in before importing data into ArrayTrack. This shows the SimpleTalk's format. Obviously, a lot of the columns that come beyond that compound column are not shown here. The full format can be downloaded from the ArrayTrack website. The columns in red, data file, hype name, sample name, label, array type, subject ID, and study title are the required columns. Everything else is optional, but the optional columns would be useful in giving the animal information, in providing the clinical endpoint information, toxicological endpoint information, so forth. So this is the type of file that a researcher would have to fill in before importing data in SimpleTalk's format into ArrayTrack. And finally, I just want to mention that if you Google ArrayTrack, you're going to get to our website, and we have a lot of resources to help you learn how to use ArrayTrack. A quick start manual is the best place to begin. We also have a set of 12 tutorials to go through each of the major functionalities in ArrayTrack. We also have a full manual that you can use. And in case you're interested in a local installation of the ArrayTrack database, you can request a CD and we will mail that out to you. Thank you.